thousands of years, humans have unknowingly shaped the genetic makeup of plants and animals through selective breeding. From the earliest domestication of dogs and cattle to the cultivation of wheat and corn, our choices have influenced the traits passed down through generations. Yet for most of history, people had little understanding of how these traits were inherited. The complexity of inheritance, where offspring sometimes resemble their parents and sometimes don't, puzzled observers. It wasn't until scientific inquiry began to unravel the mysteries of heredity that the field of genetics emerged, setting the stage for revolutionary discoveries. Geneticists are the scientists who study how traits, characteristics, and diseases are passed from one generation to the next. With advances in science and technology, geneticists have shown that genetic variation can influence disease susceptibility, and that the ability to vary genes helps species survive environmental changes. The history of genetics spans about 150 years, with research becoming increasingly specific, from populations to individuals, and finally to the molecular level. These discoveries have not only changed scientific understanding, but have also impacted medicine, agriculture, and society as a whole. Long before genetics was a science, people noticed that children often resembled their parents. The domestication of animals and plants relied on this principle, as breeders selected for desirable traits. However, the mechanisms behind inheritance remain mysterious. Selective breeding was practiced, but without a clear understanding of how traits were transmitted. This early awareness laid the foundation for future scientific breakthroughs, as curiosity about inheritance persisted through generations. In 1858, Charles Darwin and Alfred Russel Wallace jointly announced the theory of evolution via natural selection. Darwin, an Englishman, had lectured about evolution, but it was Wallace's insistence that prompted Darwin to publicly state the theory. Their work fundamentally changed how scientists viewed the inheritance of traits, introducing the concept that species evolve over time through the selection of advantageous characteristics. In 1859, Charles Darwin published The Origin of Species, formally outlining the theory of evolution by natural selection. This treatise provided a scientific framework for understanding how traits are inherited and how species change over time. Darwin's work inspired generations of scientists to investigate the mechanisms of heredity, setting the stage for the birth of genetics. Gregor Mendel's experiments with pea plants in 1865 established the concept of particulate inheritance. Mendel demonstrated the laws of segregation and independent assortment, showing that traits are inherited in predictable patterns. His publication, Experiments in Plant Hybridization, outlined these findings, which later became the foundation of classical genetics. In 1866, Ernst Haeckel proposed that hereditary material resides in the nucleus of the cell. This idea was a significant step toward understanding the cellular basis of inheritance. Haeckel's hypothesis paved the way for future discoveries about the role of chromosomes and DNA in heredity. Friedrich Miesser in 1871 identified a substance inside the cell nucleus, which he called nuclein. Later experiments revealed that nuclein consisted of nucleic acids and proteins. This discovery was crucial, as it pointed scientists toward the molecules responsible for heredity and set the stage for the identification of DNA. Lambert Adolph Jacques Quetelet demonstrated that statistical analysis could provide important insights in biology. This concept was critical to the development of biometry, the application of statistics to biological phenomena. Statistical methods became essential tools for geneticists, enabling them to analyze complex patterns of inheritance. Between 1879 and 1892, Walther Flemming, Eduard Strasburger, and Eduard van Beneden made the first accurate counts of chromosomes and observed cell division. They coined terms such as chromatin, mitosis, cytoplasm, nucleoplasm, prophase, and metaphase. Their work laid the groundwork for understanding how genetic material is distributed during cell division. In 1887, August Weismann proposed a universal theory of chromosome behavior, predicting meiosis in sex cells. Eduard van Beneden confirmed this theory, which became fundamental to understanding how chromosomes are inherited and how genetic diversity is generated. Heinrich Wilhelm Gottfried Waldeyer in 1888 applied the term chromosome to the condensed material found in the nucleus. This naming helped clarify discussions about heredity and provided a clear reference point for future genetic research. Francis Galton's 1889 book, Natural Inheritance, studied variation by quantitatively measuring differences among traits. This work formally founded the field of biometry, which uses statistical methods to analyze biological variation, and became a cornerstone of population genetics. William Bateson's 1894 book, Materials for the Study of Variation, discussed the concept of discontinuous variation, an important tenet found in Mendel's work. This idea helped explain how certain traits appear in distinct forms rather than as a continuum. Carl Pearson developed methods for analyzing statistical frequency distributions in 1894. These methods were necessary for the later development of mathematical models of evolution, allowing scientists to predict how traits would spread through populations. In 1899, William Bateson described the use of hybridization between two individuals as a tool for scientific analysis of heredity. This approach, rooted in Mendel's work, became a key method for studying how traits are inherited. In 1900, Carl Correns, Hugo de Vries, and Erich von Tschermak independently rediscovered Mendel's work. Their experiments confirmed Mendel's findings, and once they read his paper, they recognized its importance and brought it to the world's attention. Hugo de Vries in 1900 used the term mutation to describe the spontaneous appearance of new traits in evening primrose. This concept introduced the idea that genetic changes could occur suddenly, leading to new variations within a species. C. E. McClung in 1902 presented the concept that specific chromosomes are responsible for determining sex in animals. This discovery was a major step in understanding how genetic information controls biological development. Walter Sutton and Theodore Boveri in 1902 described chromosomes as unique physical entities that occur in pairs, with one member contributed by each parent. Their work led to the chromosomal theory of inheritance, which became a cornerstone of genetics. Archibald Garrod described the first human disease, alkaptonuria, that exhibits Mendelian inheritance. Later, he discussed the biochemical genetics of humans, linking genetic traits to specific diseases and paving the way for medical genetics. William Bateson in 1902 introduced key terms such as genetics, homozygote, heterozygote, epistasis, F1, F2, and allelomorph, later shortened to allele. These terms became fundamental to the language of genetics. Wilhelm Johannsen in 1903 elucidated the concepts of phenotype, genotype, and selection. These ideas clarified the distinction between an organism's observable traits and its genetic makeup, which became essential for genetic analysis. William Bateson and R. C. Punnett in 1905 performed experiments on sweet pea that demonstrated the concept of linkage. Their findings showed that certain traits are inherited together, revealing the physical connection between genes. Lucien Claude Cugnot in 1905 discovered lethal genes through experiments involving yellow mice. The population segregated in a specific ratio, revealing that some genetic combinations could be fatal, deepening our understanding of inheritance. Friedrich Laibach in 1907 suggested Arabidopsis as a model organism. This plant became widely used in genetic research, providing a simple system for studying gene function and inheritance. G. H. Hardy and W. Weinberg independently formulated the Hardy-Weinberg principle of genetic equilibrium in 1908. This unifying theory underlies population genetics, explaining how allele frequencies remain constant in a population under certain conditions. G. H. Shull in 1909 proposed using self-fertilized corn to produce commercial seed. This direct application of Mendel's scientific approach revolutionized agriculture, enabling the production of high-yield crops. Wilhelm Johannsen, while studying seed size in beans, distinguished between an individual's outward appearance, phenotype, and genetic makeup, genotype. He also coined the term gene, providing clarity for genetic studies. H. Nilsson Ale explained quantitative variation in seed coat color in wheat and oat by the interaction of multiple genetic factors. This marked the birth of quantitative genetics, which studies traits influenced by many genes. Thomas Hunt Morgan published the first demonstration of sex linkage in Drosophila in 1910. This suggested that genes reside on chromosomes, and fruit flies became a key model organism for genetic research. Alfred Sturtevant developed the first genetic map in Drosophila in 1913, solidifying the concept of linkage. Mapping genes helps scientists understand how traits are inherited and how genes are organized on chromosomes. Calvin Bridges observed non-disjunction in sex chromosomes, providing the tool necessary to definitively prove the chromosome theory of inheritance. His work confirmed that genes are located on chromosomes. Between 1917 and 1923, Calvin Bridges observed deficiencies, duplications, and translocations in Drosophila chromosomes. These findings expanded understanding of chromosomal mutations and their effects on inheritance. Frederick Twort and Felix de Heel, between 1915 and 1917, described bacteriophage, a virus that attacks bacteria. This discovery opened new avenues for genetic research using viruses as model organisms. A. E. Boycott and C. Diver demonstrated maternal inheritance for shell coiling direction in snails. Sturtevant suggested this phenomenon was controlled by the genotype of the mother, highlighting the complexity of inheritance. Alfred Sturtevant described the Drosophila bari effect, the first demonstration of position effect, and observed the first inversion in Drosophila. These discoveries deepened understanding of how gene location affects expression. J.B.S. Haldane in 1927 presented the concept of homologs based on observations of coat color in rodents and carnivores. This idea helped clarify how similar genes are inherited across species. B.O. Dodge first used Neurospora as a genetic organism in 1927. This fungus became a valuable model for studying genetic processes, especially mutations and gene regulation. J. Muller in 1927 showed that X-rays could induce mutations in Drosophila. This discovery demonstrated that environmental factors could alter genetic material, influencing the direction of genetic research. L. J. Stadler in 1928 induced mutations in corn using X-rays and established the relationship between mutation frequency and X-ray dosage. This work expanded the use of mutagenesis in genetic studies. F. Griffith in 1928 obtained transformation of pneumococci, a critical experiment that led to the eventual discovery that DNA was the genetic material. This finding was a turning point in molecular genetics. R. A. Fisher in 1930 provided the first formal analysis of selection in his book, The Genetical Theory of Natural Selection. This work integrated genetics with evolutionary theory, shaping the field of population genetics. Arne Tiselius developed electrophoresis in 1930 as a technique to separate proteins. This innovation became a key tool in molecular biology, allowing scientists to analyze genetic material and proteins. Harriet Creighton and Barbara McClintock, using corn, demonstrated that crossing over between homologous chromosomes involves the physical exchange of genetic material. C. Stern confirmed this concept using Drosophila. This discovery explained how genetic diversity arises during meiosis.